So I will talk today about introduction to FCS. And I will say that I started to work in FCS before FCS was called FCS. And I was interested in protein dynamics. And the idea was to follow a movement of a protein while they happen in real time. And of course, in order to do that, you need to be able to work in single molecules. So everything I will say is referring to single molecule, which is not the common presentation of FCS. So first of all, the idea was to go from the cuvette kind of experiment that Dave Jameson illustrated in his first lecture to number 7 in the list, which is fluorescence correlation spectroscopy. And all the techniques that you use in fluorescence, so excitation and emission spectra, you can use them in order to essentially look at the different effect on the fluctuation. Anisotropy and polarization is very important, although have not been used very much. It is possible to use in FCS. Quenching, of course, fluorescence lifetime can be done in FCS because it's essentially a measurement of occurs in a few nanoseconds. And then resonance energy transfer, which is very common, and fluorescence polarization. But all those techniques are done in a cuvette, but at some point, it was a year about 2000, we decided to go on those techniques in the microscope. And I will talk about fluorescence correlation spectroscopy, and in particular, the difference between translation and rotational diffusion. So this is very important, changing concentration and then dynamics. So let us see if I can go to the next slide. So why we need FCS to measure internal dynamics in cells? Well, the basic idea is that a method based on perturbation, like typically FRAP, fluorescence recovery after photobleaching, is very common in many labs because it's very easy to do and can be done essentially with every instrument or every microscope that you have. But there are other methods which are based on fluctuation, which are very different in nature. And I will talk about the difference between method based on FRAP and method based on fluctuation. And I say in the last sentence of this slide, there is a fundamental difference between the two approaches, although they are related as to the physical phenomena is the report. So let us talk about this idea of the number fluctuation. So if you have an open volume, which is indicated here by a red line with molecules inside, so those dots are intended to be molecules, the number of molecule or particle fluctuate according to a Poisson statistic. So this is a very important thing, that the number of particles that you can have in a volume fluctuate according to Poisson statistics. And this is due to a very basic principle of thermodynamics. So the average number that we have, of course, depends on the total concentration. So the more molecules are concentrated, the larger will be the average number in a given volume. And of course, the size of the volume. So if you have a large volume, you will have more particles and so on. The variance of this number is equal to 10, because it's a Poisson distribution, is equal to the number of particles in the volume. And this is very important because that gives us an idea how you can measure the number of particles in a given volume. But this principle does not tell us anything about the time of the fluctuation. And this is important for what I will say in the following. So if you are at equilibrium, you can have a given value for the concentration or the number of molecules in a given volume. You do an external perturbation like you do in FRAP, and then which can be positive or negative, and then you return to the value. And this return to the value is generally depends on the diffusion, depends on the physics of the process that return to the value. And this is very different when you do fluctuation measurement. So in fluctuation measurement, you have, let us say, the same average value if you observe the same volume, and you have positive and negative fluctuation. And the time the positive fluctuation or the negative fluctuation occur is random. And so here it's easy to do because you know when you do the external perturbation, then you measure the return to equilibrium. Here is much more difficult because the time the intensity will go up or the intensity will go down depends on the spontaneous fluctuation of the system as opposed to the external perturbation. So this is a table which is in a paper by Svelberg and Inori in 1911, and they wanted to show that measuring gold particles, because were easy to observe because of the scattering, that the number of the gold particle in an open volume will change according to a Poisson statistic. And that was the time when people still don't believe that in the basic things in chemistry that we have molecules, and then they were still trying to understand what is. So this is an experimental data on gold particle, and you have some numbers. This is a copy from the first page of the paper. It says 1, 2, which means I see one particle, I see two particles, I see none, 2, 3, and so on. And then you see all the sequence. And this sequence is absolutely random, so it's irreproducible. So this table cannot be reproduced in any possible way. But once in a while, you have a line here, for example. I hope you can see my cursor. And Svelberg realized that since those are random number, or random fluctuation, you can stop it and start all over again. And that means that he was silent. Simply, he was observing the sample, and then he stopped, and then he stopped again, and then he stopped again, and so on. He stopped several times, but he produced this sequence. And this sequence was produced by a metronome in which every second he was observing the system with the eyes, because there were no photomultipliers in 1911, and he was dictating the numbers to the assistant, so called as assistant. So, and this is by visual inspection, counting the particle is observed, and they were using a microscope, which today is called the spin microscope or ultra microscope, which was invented before. Now, one thing that Svelberg didn't make, he just make the histogram of those numbers in order to prove that the distribution was Poisson. But I did a little bit more than that. I simply took the numbers and I plotted as a function of time. And if you plot as a function of time, so I have the time sequence, you will observe something very, very strange. So for example, you have here two particles, and then from two particles you go to 1, but you never go to 0. And then if you observe this sequence, you will observe something which is strange, because everyone in the audience can see that is very difficult. So you have for a long time no 0 particles or something, and maybe you can interpolate with your eyes and see that actually the number of particles fluctuate with some frequencies. And this is what gives rise to the behavior of FCS. But let us see from the point of view of single molecule, or single gold particle in this case. First of all, you can plot, as Svelberg did, the number of times you observe 0 particle, the number of times you observe 1, 2, 3, 4, and then he interpolates, he shows that this distribution follows a Poisson distribution. But there is another variable that he has not done, because otherwise the field will have been very different. That is simply the time of the fluctuation. So I see 0 or 2, and then I see 1, and then I see 3. So this number of the fluctuation has to be calculated according to a mathematics which actually was not available in the year Svelberg did the experiment. And here is what I call the autocorrelation function. I will define what it is. And it shows that at very short time, so if you look at very short time, which means a few seconds after the previous observation, you see the number of particles never goes to the constant value, but goes to decreases, of course, but never goes to the constant value. So I have to explain what is the origin of this equation and how that occurs. Okay, so let us see now, sorry. Okay, let us see what we observe. So what we observe in a fluctuation experiment in the fluorescent signal, so this fluctuation, so the signal is fluctuated, is the number of fluorescent particles in the volume of observation, which can be due to diffusion and binding. And that is very important because, for example, a molecule can diffuse freely or can bind to some place. If it binds, of course, the diffusion will change dramatically. I was interested at that time in conformational dynamics of proteins, but many other things can be measured. For example, rotational motion, if polarizers are used in excitation or emission, and protein folding, blinking, which appears much later on in the literature, although we know from the experiment in FCS that there was blinking, and many more. And so that is very important because that distinguishes the physics, which is the diffusion and binding, from other processes. So let's go and see how the experiment is done. First of all, we need to create an observation volume. And the observation volume, as you can think, is very small. So the smaller it is, the larger will be the fluctuation, because the smaller it is, there can be 0 molecules in that observation volume, or 1 or 2. But I will use a language of the single molecule. So here you have an observation volume. And in the observation volume, a particle can go in. And suppose this observation volume is constant. It's not constant, but suppose it's constant. So in order to see an increase of a particle has to move from the reservoir into that volume of observation. And then eventually will leave. And this is the fluctuation in intensity, which is measured in an FCS experiment, is due to one particle, one molecule, entering the excitation volume and then exiting the excitation volume. And this is a concept which actually was not available for many years and was introduced later on. So how to obtain a small volume? Okay, so you can obtain a small volume by using 2-photon excitation. And so this is using 1-photon excitation. And that will be the excitation volume, which excites everything. And then you put a pinhole here to observe only that part of the volume. Or you can use 2-photon excitation, which gives approximately the same kind of volume. So let us see how that is done. So this is a slide that was given me by Brad Amos when he was working many years ago in this field. And that slide has been reproduced, I don't know, 100,000 of times. And every time you see something about the difference between 1-photon and 2-photon, it's something like that. So in one side, you have 1-photon excitation. This is a solution of rhodamine. This is the color. And you can see the beam going through. And you can see, first of all, that everything along the beam is excited. And yes, you can put a pinhole in some position or in any other position and decrease the volume of observation. But in this side, you have a photon which produces very short pulses ordered in time. And then you can see just one spot. And this is important because that is the spot that we will observe for illumination. And the size of this spot is due to the camera resolution was used by Brad to obtain this information. But one thing you can see is that nothing is excited around that spot. And this is very important because here everything is excited along this line, and here nothing is excited along this line. So let us see how, if we do the experiment today, what happens. So this is a real experiment in which we have beads going in solution, and then once in a while, a beads enter the volume of excitation, and you get a very large number of counts, and then it goes back to 0, and then you get a very large number of counts, and so on. So in this graph, we have 2 variables. One, for example, if we zoom in one of those peaks, we see that there is a duration, and this duration is the time a particle stays in the volume of excitation. And that duration can be, few microseconds, depending on how the particle changes. So there is an histogram of the duration time, which we call the autocorrelation function. And there is also an intensity plot. So for example, this is 2 lines. This is 6 lines and so on. And those are the photon count. But if we plot the photon counts we observe with the photon multiplier, we see that there is a distribution which is not a Poissonian distribution, but is very different from a Poissonian distribution. So we call that the photon counting histogram average, and we call that the histogram of the duration of the time. And both quantities are available in the measurement, and we will measure both quantities. So there are 2 things that we can measure. One is the distribution of the amplitude of the fluctuation, that we have to understand what it is, and distribution of the duration of the fluctuation. So to extract the distribution of the duration of the fluctuation, we use a math based on the calculation of the correlation function. By the way, the math of the correlation function was developed around 1930, 1940, and so much later than the time Svedberg was doing those experiments. So to extract the distribution of the amplitude of the fluctuation, we need a math which is based on the so-called PCH distribution. And that was developed around the year 2000 in my lab. So let us see how we can do with both of them. So first of all, what is the definition of the autocorrelation function? So this is the definition of the autocorrelation function, which depends on the time tau between 2 points in the fluctuation curve. So first of all, we have here those brackets. Those brackets means that we measure the change in the fluctuation of the fluorescence at time t, and then we multiply by t plus tau. Of course, if tau is very, very large, the fluctuation which occurs in that position does not have any relationship with the fluctuation occurring in a very long time. And then this is very important. We normalize the function by the square of the average intensity. So that bracket indicates that we do an average. So this is the definition of the correlation function. So if we know how to calculate the change in the fluorescence, we should know how to calculate the correlation function. And there was a time in the history of this field in which people used different approximation for the fluctuation and then figured out how to calculate the correlation function. So that is delta F, which is the change between the fluctuation at a given time minus the average fluctuation. So this is what is delta F. So what is the fundamental equation of FCS? Well, the fundamental equation in FCS is how the fluorescence changes as a function of time. So in order to get that, the fluorescence change because of the essentially illumination, if you illuminate the molecules or not, if you have good filters or not, depends on the quantum yield of the molecule, and then depends on this integral. So this integral says that the fluorescence intensity will depend on where the molecule is placed as a function of position. So it can be in the middle of the excitation volume, or can be out, and then depends on a term which takes into account how a molecule changes the concentration of the molecule changes as a function of time. Notice that in this equation, I have the concentration, while everything I said has to do with single molecule, which is different because the concentration is a continuous quantity and can change from 0 to whatever value, while the single molecule either is present or not. And that will make a difference in the treatment. So I say KQ depends on the instrument, but the other factor that depends on what is changing the concentration, and that depends how the volume is illuminated. So those are the terms that I will use. But if I know the volume of illumination and I know the physics of the process, so what changed the concentration, I can calculate this quantity and can calculate the fluctuation in the fluorescence signal. If I can calculate the fluorescence signal, I then can use that in order to calculate the correlation function. So now notice that the value of f of t, so the fluorescence depends on the profile of illumination, because it depends on if the particle is illuminated at position r or not. So clearly this quantity is very important, and we need to figure it out how is this going. And I will show two examples of that quantity. So this is the intensity of the volume of illumination at position x, y, and z, and I divide in two terms, one which depends only on x and y, and the other which depends on z. So notice that the volume of illumination is not sharp, does not finish here, because it's like a Gaussian. And in a Gaussian approximation, this volume will continue to infinity. So that is very important. And so it's very important also the distinction we have between one photon excitation, in which the Z is also a Gaussian. And this is the waste of the beam, is not the width of the Gaussian, but it's the waste of the optical waste of the beam, and so on. In two photon excitation, the beam decreases in Z as a function of the square of the intensity, so that has a form of a polynomial or a polynomial, 1 divided by polynomial of a Lorentzian. And so that depends, this is called the Gaussian approximation of the volume of excitation, which of course is not a Gaussian, but is an airy function, so has peaks and bands, but is different, so substantially different from a Lorentzian, a function that you obtain with two photon excitation. So that makes a very large difference in the thing. So let us see how a correlation function is. So a correlation function must go to 0 at very large time. So this is log of time, must go to 0 at very large time, because essentially the correlation at very long time are not correlated with that happens at the beginning. But at the short time, the fluctuations are correlated. So we will obtain a correlation function which is relatively large. So generally, the 0 time, of course, in a log scale cannot be measured because it will be infinitely far. So we say that the G of 0, so a time t of 0, is can be approximated with 1 divided the number of particles. And then I need to show you why that is true and how you can prove that. So that means as the time tau approaches 0, so this is nanosecond, you can go with a faster instrument, faster than that, and a long time will approach 0. So we approach a constant value at short time and 0. So a function which has a high value and then a low value will have a point in which the function decreases by 1/2. That 1/2 depends on the physics of the process, and that is called the characteristic correlation time or diffusion time sometimes. So this is what we measure. So let us go to the very first thing, is what happens at very short time. And what happens at very short time is you have to follow my reasoning, which is based on single molecule. So if sometimes I have two molecules in the excitation volume, I will have a given intensity. So if one leaves of those molecules, the excitation volume, I will have now the intensity will be divided by 2, because I have 2 and then only 1 will be there. But if I have 4 and 1 leaves, the intensity will change by 1/4 of the total. So the more particle I have, the less will be the change in intensity if one particle leaves and one comes in. And the less particle I have, more will happen. So continue the arrangement. Suppose I have 10 particles and one leaves and one comes in, will be 1 part per 10. If I have 100 particles, will be 1 part per 100. If I have 10,000 particles, will be 1 part per 10,000. So that means that the value of the G0, of the extrapolated value, due to the one molecule that leaves and one molecule that comes in, is larger the less molecules I have. And so that means that by measuring the amplitude of the fluctuation, I can measure something which is proportional to the number of molecules I have. This is very important because, for example, I can place my volume of excitation in the nucleus, and I can measure the concentration of a protein, for example, in the nucleus, or I can put in a cytoplasm or in the mitochondria, and I can measure the molecules or the number of molecules I have in those parts of the cell. And this is independent, if I measure the fluctuation, independent on the instrument, because that value depends only on the molecules leaving and coming in. And that is very important because that means that an experiment done in my lab or in any other lab in Montevideo or whatever in Buenos Aires will give this exactly the same value. So the original demonstration of that is based on the fact that you have a Poissonian distribution. So a Poissonian distribution is such that the average number is equal to the variance. So that means that the average number is equal to the variance. And let us see how we can continue. So this is the definition of the autocorrelation function, as I said. But that can be split in different parts. So this is a difference of the fluctuation. So the differential, the fluctuation, so the differential of the fluctuation to the square, it can be at time t equal to 0, of course, so tau is equal to 0, can be obtained if I have the f of t and ft average to the square divided, normalized to the square. So that will be the definition. But that definition, which I have here, if I use the variance, so this is the variance, this is the square of the average, but the variance is equal to the average in the Poissonian distribution. So G0 is proportional to 1 over n. And that is exactly the same result I obtained by saying that one molecule leaves and one molecule can add it to in the excitation volume. And this is important because that gives a very simple explanation of what is happening in the fluctuation. And when you teach this class or you teach about the thing, you can use my explanation, which is based on single model. So in order to show that that is true, so let us suppose I observe a system has one particle, none, 2, 1, 0, and this is the sequence, for example. So the average will be of this sequence will be 2.7, and the variance will be 0.257. So the n average, which is proportional to the average to the square divided the variance, will be 296. But if I increase, for example, the laser power in order to get, instead of 1, 4, whatever, intensity of 4, intensity of 8, and so on. So I take this distribution and I just multiply by a factor of 4, I get that the average, of course, increases because now the numbers are larger. The variance also change, but the ratio between the average and the remains exactly the same, 0.297. So the number of molecules is the same independent on the intensity of the laser. So now let us see about the shape of the correlation function. So the shape of the correlation function is calculated directly from the definition. If I know what is the process, so the definition was that one, if what is the process that changed the concentration as a function of time. So that the process that changed the concentration as a function of time can be diffusion. So if it's diffusion, then in that case, and I use diffusion, first of all, I say it's proportional to 1 over n. Actually, there is a factor gamma, which depends on the shape of the volume of illumination, not on the size, but on the shape. And then I have a process which depends on the diffusion constant, for example, lateral diffusion. And in this case, we have this factor 4D, which becomes 8D if you have 2 photon excitation, simply because we have the square. All the rest is the same. So if we have a 3D Gaussian excitation volume, this comes from essentially Gaussian, which is the fixed law. And then we have one more term, and every Gaussian term add a factor of square root of the term. So in the case in which we have diffusion, just diffusion, we have the g of tau will be proportional to 1 over n, and will be one term which is to minus 1 and one term which is minus 1.5, which correspond to the diffusion in 3 parts. And this is only the diffusion in 2 dimensions, and you see that this term disappears in the volume. So this is in some way very simple, and time ago I was bothering my student by doing this calculation, which is interesting to do at least once in your life, but it's very simple if you know how the concentration changes as a function of time. So suppose that we have now a different process, and this is very interesting. So for example, suppose we have a molecule is in, a molecule is not in, a molecule is in, a molecule is not in, and then I can assume that the time a molecule is in or not in, there is essentially like a lifetime. So the length of those segments there will be like a lifetime. So that means in order to account of this process, I make use of a property of the correlation function that says that since the particle is not moving, so it will be at the center of the correlation function. If the particle, its intensity will turn on and off, that means it's blinking. And then that will be the law of blink. So molecules blink, and that was known very soon as people start to do those measurements. And this is called a triple state. So the lifetime of the triple state, which a molecule will go in a triple state, so cannot be excited again, will change as a function of time. Okay, so now we use a very general property of the correlation function that says if the correlation function is due to 2 processes and the 2 processes are independent, so the total correlation function is due to the product of the 2 correlation functions. So we know the diffusion, we know the blinking, or binding, which is the same, and then we get an expression for the diffusion, which we already showed you, and the expression for binding, which is written here. So notice that binding depends on the kappa, which is the equilibrium coefficient, so free respect to bound, and then depends on the fraction of free and the fraction of bound. So in this expression, we have 2 factors, one which depends on the ratio, and the other which depends on the sum of the free and bound. So if that is true, then if you measure only binding, we can measure separately the K of the fraction of bound and the fraction of free. And this is a very general result, which is true for every process which is binary. So either blinking or whatever is given by this expression. Now, why I said all those things? Because this is a polynomial, okay, or a range of ratio polynomial, and this is blinking, which is exponential. How different are the 2 functions? Well, actually, are not very different. So, for example, I have the data which is shown in red, which is diffusion, and then I try to fit with the diffusion, which is that part, that red curve on top, and then I try to fit with an exponential. Yes, it is different, but very, very little different. So that makes that is a problem, because when you measure a correlation function, you have noise. So if you have noise here, if you have noise, you will not be able to distinguish those 2 functions so easily. So that means that the diffusion and stopping and going and binding is not really so different one from the other when you plot the function there. Okay, so there is one thing I want to say, because I want to explain how long it took for people to be able to measure fluctuation. Well, clearly it's better measured by eye, which is a way to do. So if you want to do an experiment for your students, if you have teaching a class about fluctuation, you can use the Svedberg experiment. But the point is the following, that you need 2 kinds of technique. One is how to obtain a small volume, and the other is how to measure fast. So for example, if you have a volume like a cuvette, which is on the order of millimeter, so the size, let us say, is is 10 microns or something like that, 10,000 microns. And then suppose you have 1 micromolar solution, you have on the order, according to Avogadro number, on the order of 10 to the 14 molecules. But the time it takes a molecule to cross a cuvette is on the order of 10 to the 4, which is very long time, 10 to the 4 in seconds. So, but if you have a typical cell, so which the size is about 10 micron by 10 micron by 10 micron, of course, a cell is not cubic, but that means that in terms of molecules, if a molecule is 1 micromolar solution, you will have on the order of 10 to the 5 in a typical cell. So 500,000 molecules. But the time it will take to cross a cell, because this is the calculation of the time, is only 10 to the minus 2 seconds. But if you have a confocal volume, which is what we generally use, the size is on the order of 1 micron size, you have approximately 100 molecules, which is at this point easy to measure, but it takes 10 to the minus 4 times. So you see that this quantity and this quantity is what makes the experiment to be difficult. And it took, after this experiment, about another 30 years or more to figure it out how to make a very small volume and how to measure a very fast time. So you need a fast detector and a way to measure a small volume. So to give you an idea, for example, if what happens in the diffusion constant, so if you have a diffusion constant of 300 micrometers squared per second, so that means that the Gaussian width increases per second of 300 micrometers squared, so you will have something like that. But if the molecule, and this is, for example, a small molecule in solution, but if you have a molecule like GFP, it will move about a factor, whatever, 5 times lower. Or if you have a dimer of the GFP, it will be very difficult to observe that part. So the and this equation is calculated with the D, which depends on the Stokes-Heine relationship. And the Stokes-Heine relationship says that diffusion depends on the K, which is the Boltzmann constant, the temperature, and 6 times pi, pi is a number, of course, the viscosity and that. So if you measure a difference in D, general people is attributed to a difference in viscosity, because R doesn't have a lot of, R is the radius. So, but the volume, the molecular volume depends on the cubic of the radius. So a monomer dimer is very difficult to measure. So everything was being attributed to change in viscosity. So let us see some data and let us see how it happens. So this is one of the early experiments that we did, in which we have a cell, so in another cell, expressing a protein, adenylyl kinase, which is a constant with the EGFP. So the fluorescence here is due to GFP. And those are a field, those are another field. And the difference between the two cells is simply the level of expression. Now, if we have, instead of an adenylyl kinase, we have adenylyl kinase 1, which means has a peptide added to the structure in such a way that the molecule will go in preferentially in the membrane and very little in other places. And then you can see that, yes, HeLa cell transfected with adenylyl kinase 1 beta and GFP goes preferentially to the membrane of the cell. Okay, so now let us do the following thing. So let us measure EGFP in solution, and the GFP in solution will be that yellow line here. Let us make the same GFP, assuming that it's not it's still a monomer, and then it will be the green curve. So this difference between the yellow and the green is about almost a factor of 5 or more. And people say, well, it's easy to understand because EGFP in the cell, the viscosity is different than in the solution, and then that explains that difference. Well, this is wrong. And this explanation, which appears in many, many papers, is wrong. And it's wrong because there are many people who did measure of rotational correlation time And the rotational correlation time of a GFP in solution and GFP in a cell is about the same. So if it was just due to viscosity, it cannot be that the viscosity will change a factor of 5, the diffusion, or at least will not change a factor of 5, the rotational diffusion. And that is a problem that was solved only recently when people learn how to measure in a cell smaller volume and faster in a faster time. And they figured out that actually the molecules in solution and in the cell move exactly in the same way if you look at a very small time. So in the case in which you have adenylate kinase in the membrane, you can clearly see that the process of correlation function gives 1 and maybe 2, at least 2 times. And the reason for that is because if you put the volume of excitation here or here, clearly the volume of excitation will be much larger than the size of the membrane, which is about 5 nanometers. And then you can obtain, depending where you are, a effect due to the membrane because the molecules move very slowly in the membrane to respect to what is in solution, which or in the cell, which moves much faster. But the problem is a little more complicated than that. And this is the experiment I asked my student to do. So this is the membrane, and it's about 5 nanometers, what you cannot see from this figure. But if you measure in this point, you get a 2 correlation time. If you measure in this point, you get 1 correlation time, 1 correlation time. But if you look at those numbers, all those numbers are different. So which means that there have to be something different in the different position of the cell. And this is different, well above the errors. And if you measure another line, you can see that actually at the same position at the site, it depends, it changes very much. So evidently, there are other reasons that change the diffusion constant in the cell, which is not due to simple viscosity of the interior of the cell. And that gives rise to a series of techniques that we call number and brightness, RIX, IMSD, and pair correlation function, I will talk later on, that describe how is the motion of the molecule in the presence of, let us say, obstacles in the cell or structure in the cell, which is very different than the molecule in solution. So let us go very rapidly to other things. One is example, what happens if you have 2 detectors. If you have 2 detectors and one detector, for example, is sensitive to one kind of molecule and the other detector is sensitive to the other kind of molecule, and detector 1 and detector 2, the fluctuations, see, those are different molecules, will be independent. And for example, this is 15 nanomolar fluorescence. You have detector 1 and detector 2, and clearly the correlation that you observe is very different. And that correlation is due to the fact that you have a very high level of correlation at very short time. And that is called a detector after pulse. And that due to problems with the detector, has nothing to do with the molecules at the cell. But if you cross-correlate the 2 curves, which looks at different molecules, then you can see that actually that effect completely disappears. So we know how to avoid this effect, which is due to the detector. So how you calculate the cross-correlation? Well, you get the channel I and the channel J, and then use exactly the same formula that you use for autocorrelation, but now for cross-correlation. Provide I and J are 2 different things. And when you do that, the cross-correlation calculation, of course, will measure different kind of things if you have the detector in order to measure different molecules. That is very interesting because suppose you have a molecule red and a molecule green, and the molecule which combine and form red and green will be in both channels, but the molecule which we red will only be in the red channel, and the molecule in green will be only in the green channel. So in that respect, you can isolate the molecules, as is shown here, and measure only the red, only the green and red. And there has been has been done in my lab and on other labs very early, and is possible to distinguish the molecules which are, for example, red and green. And if you measure only those molecules, you can see, you can separate the molecule by essentially destroying the bridge between the two molecules and figure it out what is the correlation due to one molecule and the correlation due to the different molecule. So, so essentially, this is what is happening. And I insist in that the cross correlation is given by this formula. And in this formula, you have to be careful that the 1 over n does not propagate to G1 and G2 and G2. So what you measure is a fraction of the cross correlation, which is due to G1 or G2, and not both the molecule. And this is, so the cross correlation experiment is a little bit more complicated because, of course, the volume of the green or the volume of the red cannot, not necessarily superimpose, and there are many different things. And the filters that you use don't separate completely the molecule. For example, the green filter, the red filter, let to pass a part of the green. So you have to take that into account. And we know how that is done, and etc. So I think, Lionel, I think my time is gone, so I will stop here.